Hello students, we are going to study about the process which is very important metabolic process, and the name of this process is glycogenesis. So in this session we are going to study about what is glycogenesis and how the pathway of glycogenesis works. The synthesis of glycogen from glucose is called as glycogenesis. The synthesis of glycogen, we are making in this process the glycogen is the end product. So the synthesis of glycogen from glucose. Glucose is a raw material for the synthesis of glycogen. The synthesis of glycogen from glucose is called as glycogenesis. Genesis means synthesis or formation. Glycogenesis takes place in the cytosol. Cytosol is the fluid part of cytoplasm inside the cell, and it requires ATP and UTP beside glucose. So generally the glycolysis takes place in the cytosol and it requires ATP. So for the continuation of the process there is the requirement of ATP and uridine triphosphate. UTP also is required. So beside glucose, so we know that the glucose is the very important factor for the process of glycogenesis, because when the glucose units forms a polymer, a chain, then only the glycogen is formed. So the glycogenesis involves following steps. There are following steps. First steps is synthesis of uridine diphosphate glucose. So the enzyme hexokinase in muscle and glucokinase in liver convert glucose to glucose 6-phosphate. There is the first reaction of this process. Then the phosphoglucomutase catalyzes the conversion of glucose 6-phosphate to glucose 1-phosphate. This is the second step of this reaction. So next the uridine disphosphoglucose UDPG is synthesized from glucose 1-phosphate and UTP by UDP glucose pyrophosphorylase. This is the first step of glycogenesis. And the next step is requirement of primer to initiate glycogenesis. A small fragment of pre-existing glycogen must act as a primer to initiate glycogen synthesis. That simply means there should be a pre-existing glycogen that should be act as a primer. It is thus primer is very essential to initiate this process. It is recently found that in the absence of glycogen primer, that means even though the glycogen primer is absent, a specific protein named glycogenin, there is another protein glycogenin, can accept glucose from uridine diphosphoglucose. So the hydroxyl group of amino acid, there is a tyrosine of glycogenin, is the site at which initial glucose unit is attached. That simply means if the primer, that is glycogen, for to initiate the glycogen synthesis, the pre-existing glycogen primer is essential. Even though it is not present, then recent study says that if the glycogen in a specific protein is present, even though the process of glycogenesis can be proceed further. So but the point is we can consider that there is the hydroxyl group of amino acid tyrosine of glycogen is the site where the initial glucose units are being attached. Next, the enzyme glycogen initiator synthase transfer the first molecule of glucose to glycogenin. So this enzyme glycogen initiator synthase, this enzyme transfer first molecule of glucose to glycogenin, that it attaches the glucose molecule to the open hydroxyl group or tyrosine amino acid, and this is the first step. After that, then glycogenin itself takes up few glucose residues to form fragment of primer, which serve as an acceptor for the rest of glucose molecule. This step ultimately says that for the initiation of glycogenesis, there is a must condition that there should be a presence of primer. For example, if the primaries glycogen primer is absent, but in that place if the glycogenin protein is present, even though this can be act as a starter or the initiator of the process glycogenesis. In the third step, glycogen synthesis by glycogen synthase. So after the initiation, the main step, that is the synthesis of glycogen, is further proceed. So here the glycogen synthase is, it is a very essential enzyme. The glycogen synthase is responsible for the formation of alpha 1,4 glycosidic linkage, the alpha 1,4 glycosidic linkage. This enzyme transfer the glucose from uridine phosphate glucose to the non-reducing end of glycogen to form alpha 1,4 linkage. So in this process what happening, the glycogen synthase is a very essential enzyme. It connects the glucose molecules with the help of 1,4 glycosidic linkage. We are aware of that in the carbohydrate, in the sugar, the molecules are attached with the help of, with a special linkage, with a special bond called as glycosidic bond. So this enzyme transfer glucose uridine diphosphate glucose to the non-reducing end of glycogen to form alpha 1,4 linkage. So the fourth step and final step is the formation of branches in glycogen. As we know that the glycogen is not a linear molecule. It has the somewhere branches. So the branches can be formed with the help of the next enzyme we are going to study it further. So formation of branches in glycogen is the fourth step in the glycogen synthesis or glycogenesis. The glycogen synthase can catalyze the synthesis of linear, so here we can see that linear unbranched molecule with 1,4 alpha glycosidic linkage. That means glucose units are joined linearly. Linearly. So this enzyme glycogen synthesis is very essential. This glycogen synthase is very essential for this process. So after that, glycogen, however, is a branch tree-like structure. I have already told you that the glycogen is not a linear structure. It has some or few branches in its structure. So glycogen, however, is a branch tree-like structure. The formation of branches is brought about by the action of branching enzyme. Another enzyme is come. So this enzyme is called as glucosyl alpha 4,6 transferase. This enzyme plays very crucial role in the synthesis of the branches in the glycogen. So this enzyme transfer a small fragment to 5 to 8 glucose residues, small fragment to 5 to 8 glucose residues from non-reducing end of glycogen chain by breaking alpha 1,4 linkage to another glucose residue where it is linked by alpha 1,6 bond. That means if suppose there is a linear chain of glucose where the glucose units are joined together, at that point, at that non-reducing point of glycogen chain, this enzyme glucosyl alpha 146 transferase, it breaks the bond alpha 1,4 linkage bond of another glucose residue, and it links to the alpha 1,6 bond. This leads to the formation of new reducing end beside the existing one. There is two ends can be formed. So next, glycogen is further elongated. Glycogen is elongated and branched respectively by the enzyme glycogen synthase and glucosyl 4,6 transferase. There are two enzymes in the formation of branches in the glycogen. The first enzyme we know that is glycogen, this glycogen synthase. This enzyme first, this glycogen synthase, this enzyme is very essential for the linear formation of linear unbranched molecule, and the another branch the branch can be done by glucosyl alpha 4,6 transferase. So this is the overall the metabolic pathway of the glycogenesis. So as we can see here that here is our molecule, that is glucose molecule. It is a glucose molecule. Okay, here is a glucose. Here is our glucose molecule. From that glucose molecule, we are going to make the glucose molecules are joined together by linearly and branching pattern, and ultimately they form glycogen. Glycogen. So glycogen is a storage form of glucose. So glucose can be stored in our body, especially in the animals, in the form of glycogen. So what happened? Glucose is converted into glucose 6 phosphate. Okay, here is a addition of phosphate group. So and this enzyme, the glucokinase, works here, and the ATP that is required for this process is converted into ADP, and the 1 phosphate that is added to the 6 carbon of the glucose molecule. So after the formation of glucose is converted into glucose 6 phosphate. After that, with the help of enzyme phosphoglucomutase, glucose 6 phosphate is converted into glucose 1 phosphate. So next step, after this formation, the UTP is converted into, as we know that the ATP and UTP are essential for the process of this glycogenesis. So then the UTP comes and it hydrolyzed to pyrophosphate, PPI. So and here is an enzyme UDP glucose pyrophosphorylase, and it forms UDP glucose, uridine diphosphate glucose molecule is formed. After that, we know that this is the first stage, and after that we require the initiator molecule or a primer, and here the primer is the glycogenin molecule, in which the OH group is free of the amino acid called tyrosine. So after that UDP is removed from this total process, and this enzyme is called as glycogen initiator synthase. After that, the glycogen primer, as we can see that some few molecules of glucose are being attached to the glycogen primer. So we have our primer is ready for the further initiation of the glycogenesis. After that, 13 UDP glucose are are hydrolyzed and to form 13 UDP. So there is an enzyme called glycogen synthase. So 13 uridine diphosphate glucose is being entered into the reaction, and only the glucose molecules are getting attached to the what we call as glycogenin, and the 13 UDP molecules are being removed from the further reaction. So after that, we can here see that the linear branching, we can see that the clear linear branching with the help of enzyme glycogen synthase. After that, there is another enzyme come glucosyl alpha 4,6 transferase. This enzyme is a branching enzyme, branching. It causes branches. So here you can see that this is the branch, this is the linear molecule, and here this is the branch, and at this point, as we can see that alpha 1,6 bond is formed. After that, the process of elongation, that means glycogen molecule is elongated and it forms more complex molecule. So elongation by the glycogen synthase forming alpha 146 bonds, and branching by glucosyl 4,6 transferase, that is the at the alpha 1,6 bond. So the molecule of glycogen is further is elongated and further branched, and ultimately it makes a complex molecule called as glycogen. and this is the process from where the glucose is ultimately converted into glycogen, that is the storage form of glucose. So this is the process of glycogenesis, that is the formation of glycogen from the glucose. This is very essential process. And when this process takes place, suppose we have a lot of glucose in our body, in the blood. At that time the glucose is stored in the form of glycogen in the liver. So this is very important topic for examination. Thank you.